work. Okay, so some examples of published work in the third section, and I sort of tried to cover the breadth of science that we see and do with our instrument. So the nice thing about being an instrument scientist here is we get to see scientists come from all different backgrounds, and so that goes from the metal community, which Daniel is part of, right up to the structural biology where we do work on proteins, and I put sort of lipids somewhere in the middle. So I have 3 examples there of work from those 3 different disciplines. So first up, the technique, small angle scattering as a technique. So small angle scattering was first observed in the 1920s on some graphite powders. So earlier in the 20th century, people began to do a lot of crystallography and designed X-ray diffractometers in order to be able to look at the structure, the atomic structure of a range of lots of different types of materials. This was all formalized by many scientists, but among them were the Australian pair of William and Lawrence Bragg. And they were first did these very simple crystal structures, and people began to do more and more complex diffraction. In 1929, 2 Indian scientists, so Raman, who went on to get the Nobel Prize, and Krishnamurti, who was one of his colleagues in India, they published a very, very short paper in Nature. So it's a letter to Nature, and it's sort of 5 very simple paragraphs where they say, we've seen this small angle scattering around our beam stop on the diffractometer. So I think the publication only has, it's 1 graph. And you can see the diffraction rings that they're interested in looking at, to look at the structure of these graphite crystals that they had. But you can also see around the big stop some intense scattering around here. And I love the fact that in the 1920s, when you saw this kind of thing, you just wrote a very short 5-paragraph thing to Nature saying, we've seen this intense scattering around our beam stop. It's not background because they've done careful background measurements. We know it's coming from the sample, and we think it has information about the sample in it. They'd already noticed that if they milled their graphite powders finer, the shape of this scattering seemed to change. So having different sized powders changed this scattering. So they thought it probably has some information in there. About 10 years after, a French physicist, André Guinier, went on to do a PhD in the area. His supervisor was a very well-known crystallographer, and his crystallographer supervisor said, there's this scattering at low angles, build an instrument so that we can measure it, which he did, and work out the maths so that we can interpret these scattering patterns so we can know what information is in the scattering. And he incredibly did over the course of his PhD. This is an extract from his PhD. It was in French, but he says here, so we noticed the existence of this very intense scattering at the center. And from this curve, from the curve of the scattered intensity as a function of angle, we can deduce the number of the characteristic of the average size of the particle that's scattering. And in some cases, we can determine the shape of its scattering. And that, obviously, the technique has advanced a lot, but those 2 things remain the same. So we can, if we have data in a good angle range, we can determine the average size of the particle. And in some cases, you can do modeling and determine the shape, the overall shape of your particle size. Just to keep on with the historical of the technique, Chadwick discovered the neutron in the 1930s, and it wasn't until a bit later, into the 1970s and 80s, that small angle scattering really took off with the advent of dedicated beamlines, both at nuclear reactors and at synchrotrons. So small angle scattering measurements conceptually are fairly simple. We take an incoming beam of a certain energy. We need to know the energy of that incoming beam. It can be X-rays, it can be neutrons. They interact with the sample, and the sample will scatter at low angles. Here it's depicted by here a 2 theta, and we will collect on our detectors. We collect a scattering pattern. The scattering patterns typically that we collect look something like this. They're fairly featureless. These are 3 examples of 3 differently shaped and differently sized particles. You can see their scattering is different, but there's not a huge number of features there for us to interpret. So it's sort of a dual-edged sword. So on a crystal, if you're trying to determine the crystal structure, you just will not get any scattering if you don't get any diffraction peaks if you don't have a nice sample. For us, we will pretty much always get a signal, no matter what you put in a neutron beam. We will measure something here, and then it's important to know, how is it to interpret this, how to interpret this signal, and also how to not over-interpret it, and not over-interpret these fairly featureless curves. And so I wanted to show you a bit today about the power. There's a power in that, and there's also some things to be wary of. Generally, to take away any maths or any equations, small angle scattering allows us to get the size of the particles that are in the beam. If things are interacting in solution, we can say how many particles are self-assembling into a larger structure. And a lot of what we do in small angle scattering is particles in solution. So often the background here will be a solvent, and these particles that we're interested in will be molecules suspended in a solvent. But not always, because of the nature of the technique. These things here, they can be voids in a solid concrete sample, they can be small dispersoids in a metal sample. As long as we have contrast between these two, between the inclusions or the molecules and the matrix, then we will get a signal. I hope everyone who's on the ANBUG mailing list knows the difference between small angle, between X-ray scattering and between neutron scattering. In that, so an X-ray scatters an atom. The more electrons that are in that atom, the more the X-rays will scatter more strongly. And so light elements don't scatter X-rays particularly strongly, while very heavy elements always do. And there's a linear correlation across the periodic table. For neutrons, of course, that's very different. The interaction between the neutron and the atom depends on the spin state of the atom. It varies across the periodic table. So some atoms will scatter neutrons quite strongly that would not scatter particularly well in X-rays. Notably, light elements like hydrogen and deuterium scatter neutrons quite strongly. There's no particular pattern across the periodic table for neutron scattering. Often cases when you won't have a strong signal in X-ray scattering. Often it's the case that you quite likely will have a good signal in neutron scattering, but it depends on the atomic composition of your sample. This is the sort of overall equation for small angle neutron scattering. So the intensity that we measure as a function of Q is equal to the number of scattering particles, their contrast, a contrast term, the shape of the object that's contributing, and times the structure factor. So if the objects are quite close together and they begin to feel each other's presence, so they're no longer just randomly distributed in space, we will have a structure factor as well that we will measure. I just wanted to say, I'm not entirely sure who is on this call, but I didn't introduce Q. So all Q is just a slightly more convenient way of expressing angle for us. So we would measure everything as a function of angle. Because we want all our data to be comparable between different instruments, we would typically always transfer that angle into a Q value, which takes into account the wavelength of the incident radiation. So everything, pretty much everything I will be showing you is represented in Q. There's an inverse correlation between Q and real space. Things that are very small in Q space correspond to large things in real space. And inversely, things out at high Q correspond to smaller length scales in real space. So contrast is a concept that's really important for us in small angle scattering. Just in the same way as optically we can't see two things that are exactly the same color and exactly the same refractive index, at small angle scattering, when you're looking at something at these distances, you can't see things unless there's a difference in scattering length density between whatever you're trying to see, the particles, and the solvent around them, or the matrix around them. In small angle neutron scattering, what we use a lot is something called changing the scattering length contrasts of samples so that we can effectively match different parts of the samples. So this is just an example here of an optical contrast where we effectively match, so that the balls that you can see when they're in air, you can see as you dip them in the liquid, which has the same refractive index as those balls, they become completely transparent. So that's called sort of an optical contrast matching. That's an exact analogy for what we do at small angles, where we will change the contrast of what we're seeing in order to make some things invisible. So in small angle scattering, what we do in neutrons more than X-rays. You can do it in X-rays as well. But what we will often do is have a multi-component system. So in this case, we've got two cats of different colors. And by changing the amount of deuterium in the solvent, we can effectively match one of the components. So for some systems here at 0% D2O, so when you have 100% H2O in the solvent, you can see both of these cats, although one slightly less. As we increase the amount of D2O, one of the cats becomes completely invisible to the eye, and we can only see one of the other cats. And it's the same as you go up to a higher amount of D2O in your solvent, you might be able to effectively match out the other cat, so match out the blue cat and only see the pink one. And I'll show you an example a bit later in the example section of how we can effectively use this technique to look at multi-component systems. In terms of how modelling data has changed over the years, I just wanted to show you an example of the type of work that was done, say, for example, in the 1950s. This is a paper from the 1950s on quite a simple protein system, and this is some more recent data on a protein system. You can see in the 1950s what people were doing is they knew that the protein was had a radius of gyration of 29.8 angstroms, so they were able to determine the radius of their scattering particle. And you can see here at the time what they would do in their scattering is go away and model a lot of different forms of ellipsoids. So they assumed an ellipsoid form of their scattering particle, and they drew up the analytical function of a prolate ellipsoid and an oblate ellipsoid with multiple different ratios between the different axes of the ellipsoids. And you can see here they then found the one that worked best and said, we think this protein in solution is quite likely to be an oblate of these dimensions. A lot's changed in computing power since the 1950s. So these days, the computational tools you have are quite incredible. For example, if you have a molecule in solution like a protein, from the scattering curves, so from these single curves, these are small angle scattering curves measured in intensity as a function of Q, you can actually determine this envelope here, this overall grey envelope, just based on the small angle scattering curve. There's an incredible power in these algorithms. Obviously, it also comes with some caveats. If you don't know exactly what is in your sample and you don't have excellent quality data, these shapes can become meaningless. So it's important to be careful. This outer shape here, which is determined by the small angle scattering, has then had the crystallographic structures superimposed. So they've interpreted the scattering also in terms of their crystallography. I suppose as a summary for what SAS can and can't do. So we can certainly always exclude a model. So we have sometimes users turning up and saying, yeah, I've got this object in solution. We think it's a sphere. Can you tell me how big this sphere is? And yes, sometimes we can say, actually, no, I can't. There's no unique model that will fit that data, but certainly your particles are not spherical when they're in solution, even though they may be when you dry them out on a microscopy slide. So we can certainly always exclude models. We can't provide atomistic information. We're not going to say exactly where atoms are in these particles. But we can get, for example, a radius of gyration. If you have data in an appropriate Q range, we can get a radius of how big your particle is. And although we don't have atomistic information, that radius is determined with high accuracy. You'll have seen even in the 1950s, they were confident to report their radius of gyration with a decimal point on it. So that information is precise when it's done well. We can also get the weight of the scattering particle, so how big the object is, which then leads to information about interactions. And in some cases, when we have excellent quality data and we have an excellent quality sample, we can determine the overall shape of the particle. The biology community, I think, have been the strongest in terms of getting together and deciding on publication quality criteria for small angle scattering. And so I'd really encourage you to read those publications if you're in that field. Versus other techniques, I suppose we might look like a bit of a poorer second cousin. This is the type of information we're getting from small angle scattering. So, for example, it's an ellipsoid. It's 100 angstroms by 50 angstroms. If you compare that to the incredible atom probe tomography people are doing, where they're ripping off individual atom by atom, and comparing it to crystallography, where we're getting incredible structures of different samples, and cryo-electron microscopy, of course, has come a very long way, and they're getting very high resolution now with cryo microscopy. So why do we still do SAS, and why are our beamlines still oversubscribed? There's a few reasons. One is the bulk. So the atom probe tomographs are incredible, but they're measuring, I don't know how small they are, but they're tiny, tiny tip for an atom probe tomography. And you're also, if you're doing crystallography, you're affected by the fact that your sample is now in a crystal structure, and that can change its structure, and we will measure things directly in the solution state if that's what's relevant for your science. I'd also add that for a small angle scattering experiment, you have incredible freedom. So once we're measuring either our solid samples or our samples in solution, we can really do a lot of different things to that sample while we're measuring it. So the measurements are on the order of minutes to hours for a small angle neutron scattering experiment. And during that time, we can be applying a whole heap of different external stimuli to your sample. So for us in small angle neutron scattering, our most popular device that we use on the beamlines is a rheometer, where we'll be shearing the solution as we're measuring the scattering from it. On an X-ray beamline, they do a lot of size exclusion chromatography, where they will get a certain amount of their solution and then run it through a column that will distribute the solution based on size of whatever you're going in. So you can really capture a substate of a complex system with these size exclusion columns. And then really, I suppose you can dream up any experiment you like. Once you're working with these things, there's no reason why we can't have light flashing to look at lumen response to light. We can do temperature. We can, if you've got a protein that's responding to salt, we can put salt in there, electric field strain, gas, microfluidics. You can do what you like, and I think that's where a lot of the power of their technique and the future of their technique is.
Hey, Katy, can I just interrupt for a moment?
Yeah.
I think your microphone is squeaking on your collar. Can you just lift it up slightly?
Oh, yeah, sure. I had the cord on the microphone, sorry.
Perfect, thank you.
Cool. So for the second section of the talk, I wanted to talk quickly about what facilities there are at ANSTO to do small angle neutron scattering, and a little bit about the instrument I'm responsible for, which is QUOCA, the small angle scattering. ANSTO is really well equipped for small angle scattering. So in Australia, we have excellent facilities to use the technique. Here at Lucas Heights in Sydney, we run 3 small angle neutron scattering instruments. We also have a lab-based SAX instrument, which gets excellent quality data, which is very much complementary to what we do with the neutrons. And those instruments are to be used together. Down at Clayton in Melbourne, you have 2 excellent SAX beam lines. The second is now operational. Here at Lucas Heights, so we run 3 small angle neutron scattering instruments. I look after QUOCA, which is here on the left, and we are next to a second small angle instrument in the guide hall called Bilby. The Q ranges of the instruments are written here, so we look at particles which are sort of up to 0.5 micron in size. Bilby has a very similar Q range, although it's designed slightly differently and can be optimized for certain types of experiments. We also have an ultra small angle neutron scattering instrument. So Kookaburra is a USANS, ultra small angle neutron scattering. So with the design of that instrument, we can get down to even lower Q values, lower angles, and we can look at particles that are on the micron length scale. Just quickly what QUOCA looks like. QUOCA is a long instrument. In the guide hall, we have 20 meters in our collimation tank and a 20-meter long detector tank. So obviously when we want to look at these very small angles, what we need to do is to be able to push our detector back to see the smaller angles. We also need to have the lower background as we possibly can, and we've put quite a bit of effort into QUOCA into reducing that background. We view a cold neutron source. So the cold neutron source is the thing at ANSTO that is currently being replaced up here. So that's within the reactor and is being replaced at the moment and should be back in the reactor by the end of September. We then, the first element of our instrument is a velocity selector bunker. And in there is what we use to select neutrons of a particular wavelength. We then prepare our beam for measurements in this collimation tank. So as I said, we're looking to measure really, really small angles in our scattering. To do that, we need a very parallel beam. So in this collimation tank, we will either have aperture after aperture along the tank in order to really have a very parallel and well collimated beam. Or we will slide guides into the beam. And when we slide guides into the beam, we're effectively bringing up more of the flux up to our sample position, but with a higher angular divergence. And so during an experiment, we will play with elements in this tank to optimize it for your experiment. The neutrons then hit the sample area, which is here, and then in our detector, which sits in our tank, we'll record the small angle scattering. Depending on the size of the particles that we're looking at, we'll move that detector up and down the tank. So currently, as I said, we're getting a new cold source. We're also replacing our velocity selector control system, which has been painful for us from an instrument scientist side. Hopefully the people coming to use our instrument haven't noticed this too much, but we're getting a new controller. This is what a neutron velocity selector looks like. So what it is a drum that rotates quickly, and basically only neutrons that have a certain particular speed will make it through these blades. So these blades are neutron absorbing. They're made of carbon fiber. They're perfectly balanced because this will spin up to 20,000 rotations per minute. And a neutron that's going at the right speed as it's spinning will make it through, and that's how we define the energy for our neutrons. We're also replacing all our guides in QUOCA at the moment. They were due for replacement. They were over 15 years old, but also we do a lot of magnetic studies on the instrument. So neutrons have a spin state, 1/2 can be, and so doing magnetic studies, often people would like to polarize the incoming neutron beam. So we want to either select the spin up or the spin down neutrons. And we do that in our guide system. Our guides were depolarizing our neutron beam, and because of that, we're replacing all the guides in our tank. We're also getting a very nice new sample changer, which I think will be something the users will be very happy with. Currently, we have a sample changer that was always very slow to change temperature. We'll now be able to change temperature within minutes rather than sort of our old temperature would take 45 minutes to stabilize, and this will be much faster. We'll also be able to improve our background yet again on the instrument with this new sample changer. So this will be a Peltier controlled device rather than an oil bath device. We're also repairing a detector currently. It's actually just finished. They've replaced this board. If you'd used Quokka in the past, you'd know we were missing these pixels here. These pixels were dead and we couldn't fix them. So the detector's been taken out and this whole board replaced, and we should be back with all of our pixels working on the detector. It's still relatively new. We bought an X-ray instrument to replace our old laboratory small angle X-ray scattering instrument. It's really quite a nice instrument that we've bought. It has a dual source. It's a Xenox instrument. We can, in many cases, match the angular range that we're getting with the neutron beam instruments. We have a USAX capability. And we also have quite a range of sample environments that we're sort of currently slowly commissioning as we get interesting science through. We've got a high temperature stage where we can get up to 300 degrees Celsius. So we can have a sample sitting at 300 degrees Celsius while we measure it. We can do more what I would call cooler temperatures with just temperature baths, and we've got a tensile stage, a shear cell, and a flow-through cell, and an autoloader for when you have a lot of samples. So not everything has been commissioned, but we're slowly getting through those things. So for the last part of my talk, I wanted to present 3 different published studies. And I was going to go from proteins through lipids all the way to metals to sort of try and cover the diversity of what we do on the instrument. So I'll start off with proteins, which was initially my background. So these are 2 users who've been coming extensively from Japan, from Kyoto University, to use our small angle neutron scattering beamline here at ANSTO. And what they're studying, one of the protein systems they've been studying is these alpha crystalline proteins. So this is a cut through of, I think it's a cow's eye, but all mammalian eyes are fairly similar. You have the light that comes through the front of your eye, and then we have a lens just behind that focuses the image down on the back of our eye, which is then taken to the brain by this optical nerve. In this lens of your eye, there's a very, very crowded environment full of a few different types of proteins. As we age, that lens becomes less flexible. Clearly, there's some molecular mechanism for that, and these scientists are trying to understand how it is that the protein structure in our eyes affects the overall behavior of the lens. There's a few different alpha crystalline proteins in there, but today I'll just talk about one, which is an alpha B crystalline protein. So alpha B crystalline, it's self-assembles. So it has these little subunits of proteins here, which are represented as little pink balls, and in solution they will associate together. When I say they associate together in solution, there's no covalent bonds that are formed between them, but they just, in solution, if you put them in water, they just like to sit grouped as together. So there's 26 of them that tend to clump together. And what these 2 scientists have come out and done with us is to look at these little self-assembled balls of proteins. And so it's a I think, relatively easy from a data interpretation. The experiment is relatively easy, but it's been quite carefully designed to, I think, optimize what we can do with the instrument. So I just tell you a bit about the Guinea analysis. I'm not sure if everyone would have not heard of this. When we do our scattering, if we have measured data in such a wide angular range that we can see your entire particle, we get at low Q, where we get what we call a Guinea region, after the famous Andre Guinea. So we will see a flattening off of these curves when they're represented on log scales, and we use this low Q region to get an overall shape, an overall size estimate of your particle. So what we do in what we call a Guinea plot, these are very standard in small angle scattering experiments, is we will plot the logarithm of the intensity measured as a function of Q squared. So this here is what we call the Guinea plot. From this log of the intensity as a function of Q squared, if you get a nice line, then you know that you're seeing your entire particle, that you haven't missed the correct Q range. And when you get this line and you do this fit, it's a fairly trivial thing to extract from it a radius of gyration. So the slope will give you this radius of gyration of your particle, and the intercept, so where this intercepts with Q equals 0, you will get I0. I0 is proportional to the molecular weight of your scattering particle, so we can say how big the object is. In the case of these type of protein systems, what we will be able to say is how many of these systems are clumping together in solution. And we will get a radius of gyration. So I introduced the concept of contrast at the start of the talk. So this is a plot which is often shown for small angle neutron scattering. So we can vary the contrast of a particle if it's in solution just by changing the content of D2O in the surrounding solvent. So the way we read these plots is this is the scattering length density here plotted on the left, and on the right is the function of D2O. So here is water in this blue line here, which is the slope. And so if we have, for example, a protein, which is here. So a protein in a completely hydrogenated environment, the signal that we measure will be proportional to the difference between this protein scattering and water, and we will get this signal. If we completely take away all of the water, the H2O, and replace it with D2O, the signal that we measure will be proportional to this value here minus the scattering from the protein. And what you can see is that at a certain water content, these two actually match. So the protein which is here, and the water which is here have the same scattering length density. So this point here is what we call our match point. If we sit a protein and we put it in 43% D2O, we don't see it at all, it becomes invisible and the scattering disappears. It's the same thing if we have a lipid sample. Different lipids will have a slightly different match point, but if you put you have lipids and you put them in about 15% D2O, the scattering signal can just disappear, which is obviously quite powerful if you've got a multi-component system and you're trying to look at one thing in that system and not the other. So we can play this game of matching out certain parts of the system. And what these scientists did, which I think is quite a clever experiment, so these self-assembled groups of proteins, as I said, they contain no covalent bonds to link these self-assemblies. They're fairly weak bonds, and they're assumed to be fairly weak to break apart. So 26 of these little balls sit together in solution. Presumably 26 of them sit together in our eyes. And what they wanted to see was if there was any exchange between different subunits. So are the proteins staying in their 26, or are they sometimes fluctuating with their neighbours and swapping places? So what they designed is an experiment where they would just mix completely hydrogenated balls of alpha B crystalline along with completely deuterated alpha B crystalline. So, and what they did was sit at the match point of the mix, the 50-50 mix between the two. So this system here in hydrogenated is completely invisible in 40% D2O. The completely deuterated version is completely invisible in 100% D2O. And if we start to mix these two, the 50-50 mix will become invisible in the middle of that at about 80%. So these are the data measured of intensity of Q, intensity versus angle, the small angle data. This is their data from the two systems. In red, we've got the deuterated system, and in blue, we've got the hydrogenated system. And you can see they've done their Guinea analysis here. So they've got a Guinea plot, which means they've got the molecular weight of what's scattering in the beam and an overall radius of gyration of the particles. And what we do when we do this experiment is we sit there and mix the two systems at the beamline, and then someone runs and presses the shutter as the person's mixing and starts the data acquisition. This is the first data set that we've got. And then as a function of time, so you can see here, this is one of the first data sets. As a function of time, what we see is the signal slowly disappearing. So we start off here, and as the contrast disappears, our signal just goes to zero. So this is exactly the same sample in the beam, but what's happening and how we explain this is that the protonated and the deuterated subunits are beginning to exchange. If they didn't exchange, the system would sit here and we would see no evolution as a function of time. And the fact that it goes down to a completely zero signal is due to the fact that there is complete exchange. So about 50% at the end of the experiment, about 50% of the monomers in these balls, 50% are protonated and 50% are deuterated. And this is their I0 plotted as a function of time, where they can then get the time dependence of the exchange of the two subunits, and they can do this as a function of temperature now. So we can determine, I think this one here is 10 degrees Celsius. So at 10 degrees Celsius, they don't exchange at all. There is no swapping of the monomers. And then as we increase in temperature, you can see it happens faster and faster. I think this is 45 degrees, and you can see within the hour everything has swapped. We're unable to capture that. And so I think this is just a nice example of how, from a relatively conceptually simple experiment and a simple analysis, we can get information on these quite complicated systems. As a second example to do with proteins, I just wanted to highlight some work that's been done recently by the structural biology community. So I think it is important to read these publications if you're at all interested in the field. So what Jill Truella did was to coordinate a round-robin study where quite a few of us participated and measured 5 different proteins across many different instruments. So we had 12 small angle X-ray instruments and 4 small angle neutron scattering instruments were involved in the study. We ended up with 247 data sets, which Jill then had to coordinate and pull all together. And the aim really was twofold. One is, what is the intrinsic reproducibility of what we're doing? So people are pushing the modeling really quite far on a lot of different systems. And so one of the questions was, well, how reproducible are our measurements? If we take the same protein from the same box and measure it on different instruments around the world, are we actually getting the same data? But also wanted to get a really standard set of data together so that when people are testing their modeling systems, they've got standard data sets to use to do all those tests with. And this is some of the analysis. It's only one graph from the publication, but you can see here these are the extracted radius of gyration and you can see along the bottom are reported for the 5 different proteins. So we chose a variety of sizes, so they had a very small protein, which is RNAs, right up to some of the larger ones. And from that data, they extracted in each case one of these radius of gyration and a maximum dimension of the particle. And all these different black points here are the different facilities around the world. And the red is the average radius of gyration with a reported standard deviation. So you can see, as I said, the technique, although we don't get atomistic information, we do get, for example, this parameter of a radius of gyration, we do get it with a very high degree of precision. The standard deviation on that is small. It's within 0.1 of an angstrom. There is intrinsic, you can see for some proteins there was a wider, a larger standard deviation in the measurements. So for example, this sample here, which was lysozyme, just inherently seems to have a larger distribution of parameters. And one of the reasons for that is we think it's very much that the samples, as we make them, are not as reproducible in this case. So, but we did, at least certainly we all were very confident that all of our measurements matched up very nicely, with the exception of a small offset for background between them. So the background subtraction seems to not be perfect in all cases, but these data sets are now in their database. So another thing that the biology community is doing a good job of is publishing curves in a database, which you can then go in and download and use for your own research. Oh, I had an example on lipids, but are we running out of time? Daniel, did you want to leave 10 minutes for questions?
Yeah, but I think it should be fine.
So I'll just do the lipids. I'll skip the metals. People can ask you about metals.
Oh yeah, yeah, it's fine. Yeah, sure.
So I just talk a little bit. Lipids is another area that we do see quite a lot of work on our instruments on. So a good chunk of our user base is probably people looking at self-assembly in solution of various amphiphiles. So molecules that are hydrophilic in one part of their molecule, and then they'll have a hydrophobic part as well. What happens, just like when you have detergent going into solution, it forms quite a variety of structures, so quite a rich structural landscape. You can get very small spherical micelles, you can get larger structures, you can get lamellar phases where they'll line up in lines to hide these hydrophobic groups from the hydrophilic exterior. Because there's some order in these systems, but there's not a high degree of order, they're not amenable to crystallography, and we're still learning quite a lot about the structure of these molecules in various solutions. One of the basic functions of these molecules is they form the barrier on the outside of all cells. So all of the cells that are in our body are made up of a membrane, and a very big component of that membrane is lipid molecules. You can see here represented, that was artistic, but it also gives you some idea of what a biological membrane looks like. There's a lot of different proteins in there. The proteins are these green blobs, so some of them go across the membrane, some of them stick out from the membrane. And holding this whole structure together are these lipid molecules, which have a hydrophilic part nearest the water and then a hydrophobic part in here. And this makes the barrier within our cell. There's a whole range of different lipid molecules, and as I said, we're still learning a lot about them and their structures. One nice thing you can do with lipids, if you buy a solution of them, where you buy them in a powder form and then you can make up a solution, is relatively simply, there's relatively simple lab apparatus where you can make up spheres, basically hollow spheres of these lipids. So you can get different mixtures of lipids, put them together, put them in a buffer and make up a nice sphere. So you push, in this case, we're pushing it through a membrane with holes in the membrane of defined diameter. And as we push the lipids through these holes, you can get a more and more homogeneous system of lipid spheres. So we call them liposomes. In this case, I've been playing around with two different types of lipids, so a DMPC and TMPG. One of them is a charged lipid and one of them has no charge. So this is an example of a molecular dynamic simulation of what a lipid membrane might look like. You can see it's incredibly disordered in there. This is an example of some small angle scattering data where I've made these quite large little hollowed lipid spheres. So, and from this data, we can get a radius of the overall sphere and how thick the membrane is. So this is, these curves have been fit over the entire Q range with only those two variables, a radius and a thickness. And you can see as we change temperature, we get changes in this structure. This is an extraction of the membrane thickness of the liposome as a function of temperature. You can see there's a change from lower temperature to higher temperature. The membrane actually becomes thinner. This is due to the fact that at low temperatures, these lipids tend to form as elongated straight chains in their chains, in their hydrophobic chains tend to be quite rigid, and as at higher temperature these chains melt and you get this a softer system where the chains are more flexible and they will actually squeeze the membrane down into a thinner membrane system. So what happens with temperature is that the thickness of these liposomes actually decreases, and this is also affected by charge. So this the charges in the head group, as you increase the charges, we can tell that there will be a greater repulsion of those outer leaflets, and the thickness becomes greater as well. This is partially compensated by the fact that the entire structure is changing in shape. So as this thickness thins out, the radius of the scattering particle needs to increase, and we see a larger overall liposome. This is also a fun system because we can do dynamics with neutron scattering. So some of our users are using both small angle neutron scattering and inelastic techniques at ANSTO, and so we can then, there's a nice simple system that we can do these dynamics measurements on, for example, on Pelican, which is pictured here. I just wanted to say, so the systems I've been working with, these liposomes are relatively simple, but as we've, since the advent of mRNA vaccines, which were used in our COVID vaccines, we've had a real increase on the beamlines in people doing work on lipids on much more complex systems than what I showed. So this is a lipid nanoparticle. At some work. This wasn't done with us. The work that's been done on Quokka is yet to be published. But this is the type of thing people are doing with these lipid nanoparticles. So the lipid nanoparticles that are in COVID vaccines are, the mRNA is stabilized by a number of lipids. So they seem to work well with a mixture of lipids in them. I think in this one there's 3 lipids. There's a pegylated lipid that forms at the surface that seems to control the size of the overall nanoparticle, and there's cholesterol, which is also in there. And what you can do by very carefully deuterating either the lipids or the cholesterol is to determine the overall distribution of the lipids and cholesterol in the nanoparticle. And so you can see here the type of information they get on these nanoparticles is the core of the nanoparticle, how big it is. They can determine that the cholesterol seems to cluster in the outer ring of the nanoparticle. Let's put that in. I'll skip metals. I'm sorry, I was longer than I thought. I'll jump to my conclusion. Oh. Get there. So as I say, it's an excellent technique for the nanostructure, and there's a huge variety of experiments that you can do with the technique. But if you don't know your sample well, so on a random sample that you just come and say, can you do some small angle scattering on this sample and give me information, you probably will get very little to no information from it. The data modeling has come a long way, and I would say the biology community is really leading the way in terms of making sure that there's standards in place for reporting your data and making sure you're not over-interpreting the data. I just want to thank all my colleagues. So these are the group of small angle scattering scientists at ANSTO. We'd love to see you on our instruments. The next proposal deadline is in September. And I've stolen slides and pictures from various people. So I just wanted to thank them, and there are a few references. Thanks for your attention.
Thank you, Katy, for this very nice presentation. So anyone who has questions can unmute yourself or type your question in chat. So is there any question for Katy?
Hello, I have one question.
Yep.
Thanks for that. That was great. So I noticed that you changed the so obviously a big part is that you changed the contrast using water and deuterated water. With many proteins, water plays a huge part in terms of the self-assembly. So for Alzheimer's, for example, amyloid fibers start off as a liquid-liquid phase, coagulate, and then depending on how the water interacts, they end up forming hydrogen bonds. Does the deuterated solvent, could that not alter the results that you're getting through the small angle scattering?
Yeah, it does in some cases. So in some cases we're lucky and it doesn't. In some cases it does. So the work, for example, when they're doing these mixing experiments on hydrogenated and deuterated things, we check quite carefully with small angle X-ray scattering. So we'll do first a sort of a series of measurements in H2O and D2O just with X-rays to check that that's not changing the fundamental structure of what we're seeing. Sometimes it does affect it. And sometimes there's things you can change in by adding in, for example, salt to change the water composition. We would love to say there is a pattern to why some proteins seem to be okay with D2O and some proteins seem to not like it. We haven't found that yet, but something actually one of my colleagues was trying to do something a bit systematic in this space so we can get a feel for, well, when is it actually going to be a problem? And we're not, because for now we're testing this independently. So we'll run SACs both before and after the experiment generally to try and be certain that things are stable and that the deuteration hasn't affected it. In some cases, unfortunately, the effects can be large.
That's a bit of a shame. Thank you.
Yeah, so I haven't seen it be very large for protein systems. I've seen it on these what do you call it? These microemulsions. It had a very strong effect on some microemulsions we were looking at.
Thank you.
I have also a question.
Yep.
It's mainly about the, let's say, the complexity of the system. You mentioned that you measure mainly the shape of relatively large particles, not the atomistic structure.
Yep.
So in the case of the one, for example, that agglomerates into 26, 26 of the proteins agglomerate in a larger.
Yep.
What happens if you have a system that is not very consistent in terms of the shape? Let's say it can form 10 and 50 or a different size of particles. Will you be able to see only the average particle, or can you do something else to kind of make it a bit more homogeneous?
So it depends, is one answer. But so say we have a mix, we would try to really push the modeling as far as you can. Then you would want to get rid of these larger particles to make sure you're only looking at one particular type of particle. So if your object is nice and monodisperse, you can really push the modeling quite far in terms of these, what they call ab initio methods. if you can just have one subspecies. If you have two species in solution and you know it and you can fit your data to two different populations, then you can still get information from that. You always see an average, and the average is weighted by the size of those different mixtures in solution. So, for example, the higher molecular weight ones will actually contribute more to your scattering. So it's not it's not just the average of the two, it's an average weighted by other factors, and one of those factors is their weight. So the larger ones will contribute more. If we can get a nice monodispersed solution, we definitely prefer to work with that. At the synchrotrons, they use a lot of those size exclusion columns where they will separate out the protein by size, and so that you're only looking at one particular species in that solution. Because of we don't have the signal to noise that a synchrotron does, we tend to not be able to do that with the neutron work. But most people who come to use our instruments on anything that's solution based, they will mix it up immediately before, and in the case of the protein people, they will often run it on a column just before and then take it straight from the lab to the instrument to measure it as quickly as possible before any of those populations have changed.
All right. We can have one more question. Anyone have questions? Julius?
Katy, thank you for the talk. So I just have a question regarding the solvents. So are there any specific solvent needed for SAS, or is it suitable for all solvents, including both aqueous and non-aqueous?
Yeah. So so there's no particular restriction on the solvents you can use. I have a commercial group that come to use the beamline a lot, where they're using all sorts of organic solvents, actually. And that's one example where some of those organic solvents don't make SACs quite so amenable. So they will specifically come to use neutron scattering when they have certain particles that are only soluble in solvents, where they know they will get a good signal in the neutron small angle neutron scattering, and they can't do X-rays on those. So there is no limit on the different solvents.
Oh, good. Just a follow-up question. So how do you handle liquid? How do you handle liquid samples that are air sensitive?
Have I done air sensitive work? You can seal your Helmer cells in a glove box if that's suitable. You can sometimes people come with things already sealed in their own sample holders if that's something you can do. As long as we can send you a sample holder, you can fill it and seal it in either your laboratory or you can use our glove box here, then there's no reason why we can't do air sensitive samples.
Oh, good. Thank you, Katy.
But happy to talk to you offline about that, Julius.
All right, thank you.
Yeah, thank you. So for anyone who has more questions, can contact Katy after this seminar. Yeah, thank you, Katy, and everyone who coming to this seminar. I hope you all enjoy this event. We have more future town hall events and seminars in the future. I hope you all enjoy it. Thank you, Katy. Thank you,
Thanks,
everyone.
Daniel.
Thank
Thanks,
you.
everyone. Bye.
Bye.